Okay, Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Nazrin, and today I want to talk about diversity of marine phototroph. So, let's go for the first slide. So, for the first slide is about traditional understanding of primary producer. So, the primary producer in marine mainly the diatoms and dinoflagellates. But why? Why are only these two? So, this is because the major organism that can be collected by the phytoplankton net and widely distributed is these two lah. Maksudnya only these two yang widely distributed and can be trapped by our phytoplankton net technologies at that time. So sebab phytoplankton ni dia ada only 3 size, which is 100, 70 and 60 at that time. So in other words, phytoplankton that below this size can pass the net easily. So but however, recent phytoplankton research using deoxyribonuclease, which is DNA detection and flow cytometry, found out yang there are predominant phytoplankton that is smaller than the phytoplankton net can catch. Maksudnya ada lagi phytoplankton yang below this size lah, yang below size yang phytoplankton net can handle. So for example, haptophyte including Phototroph. This is because Phototroph ni their size dia is below than 50 micrometer. Okay, so for the second slide is about recognition of Picocyanobacteria dominance, which is from 1970 to 2000s. So because of the development of observation technique, found out yang ada species named Syncococcus species that are dominantly distributed in the marine environment, especially in tropical and subtropical regions. So the species also play significant role as a primary producer dalam this environment. So in the late 1980s, ada development of the high performance liquid chromatography and technique for pigment analysis and cell detection technique using flow cytometry yang membantu dalam research of this Picocyanobacteria. So they found out yang Syncococcus ni contain di vinyl chlorophyll A yang ada absorption spectrum yang berbeza daripada usual chlorophyll A, as dia tak ada, as it did not contain phycobilipigment yang sepatutnya ada dalam Syncococcus biasa. So apa itu phycobilipigment? Phycobilipigment ni ialah light harvesting antenna dalam common Syncococcus yang boleh dijumpai dalam Syncococcus yang biasa. So the next cyanobacterium is Phlorococcus. So Phlorococcus ni can perform oxygenic photosynthesis using DV-CHL A atau B yang boleh efficiently absorb the blue light and dominating the ocean. So the amount of DV-CHL A dalam laut kita 3 times higher than usual CHLA. So ini saintis membuat kesimpulan mengatakan yang Phlorococcus ni also playing as a significant role in our marine environment, which is as a primary producer. Okay, the next slide is discovery of ubiquitous photoheterotroph from the year 2000 until the current time right now. So there are two findings yang stating that the microorganism other than traditional phytoplankton could possibly contribute significantly to the transformation of light energy into chemical energy in marine environment. So there are two two bacteria yang they found out, which is the aerobic and oxygenic photosynthetic bacteria, or AAPB in a short form, then proteohodopsin containing bacteria. So I want to talk about APB first. So APB was derived from non-sulfur bacteria, which is anaerobic and oxygenic photosynthetic bacteria, or in short form NAPB, in the alpha proteobacteria. So NAPB ni perform photosynthetic in anaerobic environment, which is anaerobic environment is the environment without oxygen. So APB ni the same with NAPB, but at the point yang they contain BCHL, that part is the same, but APB are in aerobic environment, but NAPB in the anaerobic environment. And APB ni memerlukan molekular oksigen untuk grow dan BCHLA synthesis. Kalau tidak, dia tak boleh bekerja lah, sebab dia bekerja dalam keadaan memerlukan oksigen. So for the next one, so AAPB ni boleh dijumpai dalam euphotic zone ataupun zone yang di mana berada pada paras permukaan lah. Maksudnya in the surface of the marine environment. So that's where you can find the APB. So for the next slide is they had been reported to grow faster in seawater other than heterotrophic bacteria. But based on the article, the physiology role ni was not clarified yet, tapi they must contribute to the transformation of solar energy in marine environment and also act as one of primary producer. And the next one is proteorhodopsin. Proteorhodopsin is a well-known is a rhodopsin-like protein detected in marine proteobacteria. And rhodopsin ni dia bertindak sebagai photoreceptor ataupun light sensor. The one rhodopsin ni was known to contribute to the ATP synthesis, di mana bakteria rhodopsin tadi bacteria rhodopsin transform light energy to the transmembrane proton electrochemical gradient for ATP synthesis, and di mana dia menukarkan tenaga cahaya tadi untuk dihantar kepada transmembrane proton electrochemical gradient untuk ATP synthesis. And the total amount of proteorhodopsin containing bacteria ni ialah about 13 to 80% of the total amount of bacteria and archaea in marine environment. So boleh dikatakan ia memainkan peranan penting in light energy transformation in the marine environment. And light dependent ATP synthesis ni was reported in Flavobacterium strain, but not all proteorhodopsin containing bacteria has this kind of light dependent growth. And proteorhodopsin also advantages for recovery from starvation other than for ATP synthesis. But how? Based on my research, it was said that proteorhodopsin provide a physiology mechanism that allow surface ocean bacteria to manage an environment where resource fluctuate markedly. Maksudnya ada naik turun resource, tapi dia menyediakan physiological mechanism. Okay, so the next slide is about oxygenate photosynthesis using far-red light from years 1990 to 2011. So there are three chlorophyll that I want to introduce to you, which is CHLA, CHLD, CHLF that I will use in this slide. So CHLA is dispensable for oxygenate photosynthesis, where it plays essential role in photosynthesis process, as we all know. And so the in situ light absorption range by CHLA is about 400 to 700 nanometer, and is defined as photosynthetically active radiation, or in a short form is a PAR. So PAR ni digunakan untuk evaluation of light strength ataupun light intensity for the activity of oxygenic photosynthesis, di mana dia memerlukan dia mengukur kepada kekuatan cahaya untuk aktiviti oxygenic photosynthesis tadi. Dan ada satu cyanobacterium yang tidak menggunakan selain daripada CHLA, which is CHLD. CHLD ni menggunakan CHLD ni ialah red shifted chlorophyll that can absorb far red light 30 nanometer long than CHLA, which is from 700 sampai 740 nanometer. The name of cyanobacterium is Acariochloris marina, and it has the equivalent energy transfer efficiency, which is equivalent to CHLA. And the next slide, in 2010, another red shifted chlorophyll CHLF was reported. So CHLF ni dia ada absorption maximum at around 720 nanometer, and it can absorb far red light up to around 716 nanometer. So it is more than CHLD and CHLA tadi. But at that time, it can be taken into account as it need further research need to be done for it to be applied in our marine study. Okay, so for the next slide is discovery of pico-eukaryotic phytoplankton. So eukaryotic phytoplankton ni has been revealed to be widely distributed in significant amount in marine environment, but only some dozen of species in Chlorophyta Trogontophyta and Haptophyta yang dikatakan ada dalam menjadi eukaryotic phytoplankton until the early year of 2000s. And by pigment composition analysis of marine water, carotenoid 19 or hexanoloxyfucazontin atau short form ni 19HF was predominantly detected in the picoplankton. And Picocyanobacteria, which is well known as a predominant phytoplankton, do not contain this 19HF as it is specific pigment in the Haptophyta. So the amount of phytoplankton in Haptophyte is around 50%, and Picohaptophyte form 5% of the global phytoplankton biomass and contribute to primary production. In 2007, there are also phytoplankton that does not belong in any phylum, but the sequence was close to the Cryptophycean lineage and was named Phycobiliphyte. And in 2011, they found new candidate phylum and was named Rhapomonads. So Phycobiliphyte ni widely distributed in coastal areas of Europe and North Atlantic, while Rhapomonad is in coastal area of Europe, Atlantic and Pacific Ocean. So the next slide is strange phototrophic microorganism. So the cyanobacterium light particle CyanY was detected by metagenomic analysis of nitrogen fixing bacteria in marine water. It was first detected as a cyanobacterium which had a nitrogenase gene for nitrogen fixation in pelagic water. So usual cyanobacteria grow by an oxygenic photosynthesis process photosystem and genes for carbon fixation system, but UC1A lack a photosystem 2 which needed for splitting water into proton, electron and molecular oxygen, fixing CO2 and so on. So the lacking of the essential gene suggests that UC1A might be a symbiont. So what is symbiont? Symbiont is like living together with other organism. So since it possesses a complete gene set for photosystem 1 and contain chlorophyll, it must use light energy for some activity of the particle. So organism that can grow using light as its energy sources called phototroph, whether it is photoautotroph or photoheterotroph. So photosynthetic or synthetic organism are the representative of phototroph as it can undergo photosynthesis process. Photosynthesis process can reduce carbon dioxide into organic compounds such as sugar. And primary producer are organism that synthesize organic compound using inorganic carbon. Maksudnya, photosynthesis or synthesis process reduce carbon dioxide into organic compound, and while primary producer reduce synthesize organic compound into inorganic carbon. So they are quite the same thing, but recent discoveries tell that it is not suitable for the term to be used is not suitable because not suitable for the understanding of carbon and energy workflow. So in a chloroplast or cyanobacteria, proton are pumped into thylakoid lumen from stroma, while in non-oxygenic photosynthetic bacteria, proton are pumped out from the cytoplasmic space to the periplasmic space. This is proton pump into or out must be through ATP synthase, which result in synthesis of ATP. So primary producer are not only the organism that produce organic compound from inorganic carbon, tapi ia juga sintesis ATP daripada tenaga matahari. The last one is research timeline. So in 1970, phytoplankton are mainly diatoms and dinoflagellates. Then in 1980, development of HPLC technique found the Picocyanobacteria, and in the year of 2000, they found two widely distribution microorganisms, which is AAPB and Proteohodopsin. And in 2010, new finding of phytoplankton based on metagenomic analysis and flow cytometry. And the last one is 2011, they found Rhapomonad was found when using metagenomic analysis. Rhapomonad is a new candidate phylum and was found using metagenomic analysis. Thank you.